This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
Okay, good muggy afternoon, everyone. I hope you're all comfortable in here. It's nice and cool. So I'd like to thank you for joining us on the 7th talk of this Grid Talk series, which is sponsored by the UCSB Office of Summer Sessions in collaboration with the Alumni Association and the Carsey Wolf Center. So today I have the pleasure to introduce Dr. Dennis Clegg. Dr. Clegg earned his bachelor's in science degree in biochemistry at UC Davis and his PhD in biochemistry at UC Berkeley, where he used emerging methods in recombinant DNA to study the sensory transduction systems of bacteria. As a Jane Coffin Childs postdoctoral scholar at UC San Francisco, he studied neural development and regeneration. He has continued this avenue of research since joining the UCSB faculty, with studies of extracellular matrix and integrin function in the developing eye. His current emphasis is in stem cell research, with a focus on developing therapies for ocular disease. Dr. Clegg is the recipient of the UCSB Distinguished Teaching Award in Physical Sciences, the UCSB Community Affairs Board Award, the National Eye Institute Audacious Goals Award, which is pretty awesome in my opinion, and served as a chair of the Department of Molecular, Cellular, and Developmental Biology from 2004 to 2009. He has been a Frontiers of Vision research lecturer at the National Eye Institute, a keynote lecturer at the Stem Cells World Congress, and a TEDx speaker. He is founder and co-director of the UCSB Center for Stem Cell Biology and Engineering, and serves on advisory boards for the California Institute for Regenerative Medicine and the National Institutes of Health Center for Regenerative Medicine. He's also a co-principal investigator of the California Project to Cure Blindness, which is a multidisciplinary effort to develop a stem cell therapy for age-related macular degeneration. So please help me welcome Dr. Dennis Clegg to the stage.
All right. Thank you all for coming, and thanks to the organizers for the invitation to speak to you tonight. I'm going to tell you a little bit about stem cells and the different kinds of stem cells. I'll start with that, and then I'll go into the ways that we're using stem cell research to develop cellular therapies for ocular disease. And the title of my talk is Vision for the Future, Stem Cell Therapies for Eye Disease. Now, in preparation for this talk, I had to come up with some images. So, I went to the web and I searched for cell therapy, and the first thing that came up is this. It's a shampoo. Anybody use cell therapy out there? I don't know if it's a good shampoo. That's not what I'm going to be talking about. I'm going to be instead talking about developing therapies for disease using cells. And some have called it the third pillar of medicine. We have small molecule drugs that we take a pill or get an injection. And then we have biologics, things like antibodies and growth factors that are injected into our bloodstream. Now what people are looking at is using cells, a much more complicated sort of therapy, but actually a much more intelligent type of therapy where you inject a small molecule and it just goes through the bloodstream and does its thing, kind of unidimensional. A cell is a complicated living thing that can make tissues and replace tissues that are damaged or dying in disease. It's sort of like if your car wasn't functioning well and your spark plugs weren't working, you could add an additive to the gasoline, like a small molecule drug that would make them work better, or you could replace the spark plug, build a new one, and put it in, sort of akin to using cells to replace body parts that are damaged. Okay. So part of this is stem cells, and those are the cells of choice for cell therapy. What is a stem cell? Well, I'll tell you the answer. Two things that the cell has to do to be a stem cell. First, it has to be able to replicate, make more copies of itself. And then second, given the right signal, it differentiates or changes into a range of specialized cell types. And there are several types of stem cells. Pluripotent stem cells can give rise to most cell types in the body, and multipotent stem cells can give rise to a few cell types. So examples that I'll talk about are embryonic stem cells and IPS, or induced pluripotent stem cells. Those are the first category, and they're very powerful. They can make just about every cell in the body. Adult stem cells fall under the second category. They're multipotent. So an example there is a bone marrow cell that can make all of the blood cells, but can't make neurons in the brain, for example. First, let me talk about embryonic stem cells, and let me address where do embryonic stem cells come from. There's a bit of controversy associated with this, and to understand, we have to go back to basic developmental biology. How many have taken developmental biology? Raise your hand. Okay, very good. That's not very many, but when sperm meets egg, you create a zygote, and then it divides, goes from one cell to two cells, then two cells to four cells to 8 cells, etc. And after about 4 days, you develop what's called a blastocyst, and it's a little round sphere of cells. And if you look at the picture in the bottom right, you can see a cluster of cells sort of on the ceiling inside that sphere. That's called the inner cell mass. And at day 5, that appears. And that is a really special class of cells. The ICM, or inner cell mass, is what develops the entire embryo. So that sort of nondescript cluster of cells develops into the embryo. The other cells on the outside make the extra embryonic tissues like the placenta. At day 6, then, you have implantation into the uterus. Now, embryonic stem cells come from the inner cell mass of a 5-day-old blastocyst, and they discovered this working in mice back in the 80s and found that if they go in and pull these cells out, just like what's shown in the picture here, and grow them, they can actually make an entire mouse from one of those cells. So they're pluripotent, and they called them embryonic stem cells. Now, in humans, where do we get these? Excess blastocysts, it turns out, are generated during the process of in vitro fertilization. And rather than throw them away, some couples have chosen to give them to science and use them for research purposes. Now, it took a while to figure out how to make the human embryonic stem cells. James Thompson at the University of Wisconsin, back in 1998, published a paper in Science showing for the first time that you could make these cells from human tissue. And it took a long time to figure out how to do this. And here's a picture of Jamie on the cover of Time magazine, the man who brought you stem cells, in 2001. Now, Dr. Thompson has a relationship with UC Santa Barbara. He's actually part-time at UCSB, and he's a co-director, along with myself, Tom So, and Pete Coffey, of the Center for Stem Cell Biology and Engineering. And we don't have a medical school here, but what we're trying to do is combine basic studies in stem cell biology with bioengineering and come up with novel discoveries that can then be translated into clinical practice. So Dr. Thompson has a satellite lab here. His main lab is in Madison, Wisconsin. We have noticed that he comes to Santa Barbara more often in the winter, and I'm not sure why that is, but here's a picture from his first publication in 1998 showing that he could make human embryonic stem cells. And they're difficult to grow. They grow as a colony, a cluster of cells, and you can see in panel A in the upper left-hand corner a colony of thousands of cells. And they don't like to grow as single cells. In fact, in these early experiments, they had to put a carpet of irradiated mouse embryonic fibroblasts down on the surface of the plate and then add little chunks of colonies that would grow up and form these structures. And it's almost an art form to figure out how to grow these. You can't just read a paper and go into the lab and do it. And in fact, in preparation for this talk, I found evidence that it is indeed an art form. Here's a picture of embryonic stem cells in culture with Vincent van Gogh's Starry Night. Maybe Vincent van Gogh was a stem cell biologist. We don't know for sure, but there you go. Okay, so that's embryonic stem cells. Oops, too fast. Let me talk next about this exciting new class of stem cells called IPS, or induced pluripotent stem cells. And maybe you've heard about this, but back in 2006, Shinya Yamanaka in Japan discovered that he could take mouse tail cells, just skin fibroblasts, and convert them to what look like embryonic stem cells by adding in 4 genes. And they're called OCT4, SOX2, KLF4, and MYC. And these are genes that are normally turned on in the embryo, but they're turned off in the adult. So if you turn them back on in the adult cell, it reprograms that cell to think it's young again, rejuvenates that cell, and turns it back into an embryonic state. Then in 2007, on the same day, Yamanaka and Thompson reported independently that you could do this in human. You could take a biopsy of skin, add 4 genes, and turn it into an embryonic stem cell. And they called this IPS, or induced pluripotent stem cells. You can now do this from blood and many other tissues besides skin. And interestingly, Yamanaka used one set of factors. Thompson used a slightly different set of transcription factors, and both seem to work to generate cells that are pluripotent. They can make almost every cell in the body. This is very exciting for 2 reasons. One is now you don't need embryos. You can start with skin. And second, if you're starting with a patient with a disease, you could make a cell that, at least theoretically, would not be rejected by the immune system. It would be recognized as self and could be used in therapies. Another exciting advance that has happened is using IPS cells to study disease mechanisms. Let's say you have someone with an eye disease and they go blind, but you don't know what is going on in the photoreceptors, the rods and cones. They just die. Well, you can't really go in and pull out rods and cones and study them, but what you can do is take skin from the patient, convert it to an IPS cell line, and then convert that IPS line to the cell of interest, say photoreceptors, in culture, and study those now in a dish in unlimited quantities and try and figure out what's different about the patient's photoreceptor compared to a normal photoreceptor. And maybe then you could screen with different combinations of drugs in a petri dish and find drugs that convert the abnormal back to the normal. And they call this disease in a dish. It's a way to study a variety of diseases from Alzheimer's to cardiac disease to all kinds of different diseases that we don't understand. So very important advance, and lots of studies going on, particularly here at UC Santa Barbara. Studies of ocular disease and neural disease are going on using this approach. And of course, there are lots of different non-embryonic stem cells, so-called adult stem cells, that are present in your bodies. In fact, the most common stem cell in your body is actually the adipose stem cell. In fat tissue, for unknown reasons, there are a lot of stem cells. They're multipotent. They can make about 4 or 5 different cell types, but they can't make brain cells, for example, or ocular cells. People ask me all the time, when are we going to have stem cell therapies? Why is it taking so long? Well, we've had a stem cell therapy since 1955, when bone marrow transplants were first developed. And I mentioned earlier, the bone marrow contains a hematopoietic stem cell, a cell that can make all the different cell types in the blood. And people have been transferring that from patient to patient for a long time. Another area where you see stem cell transplants in the clinic today is in the cornea, on the surface of the eye. You can transfer corneal stem cells from one patient to another, or from a donor eyeball to a patient, to regenerate the cornea. So we already have some, and some use adult stem cells, and more are under development. So which cell is best? Embryonic, iPS, adult? And the answer is it depends what you want to do. If you can make your cell from an adult stem cell, that might be fine to start with an adult stem cell. But some cells, we just can't seem to figure out how to make them from adult stem cells, and the cells are not capable. So in that case, you need a more powerful cell, a pluripotent stem cell. And really, more research is needed. We're in the infancy of this kind of research now, and more work needs to be done in this area. Okay, why is everybody excited about stem cells? Well, you can imagine replacing parts that you lose or that get sick by making a tissue in a petri dish and then putting it in to replace dead or dying cells. Things like Parkinson's disease, Alzheimer's disease, diabetes, spinal cord injury, heart disease. And if you're interested in a particular disease, the California Institute for Regenerative Medicine has a website where you can click on a disease and see the type of research being funded in this state by our wonderful state-funded stem cell research program run by CIRM, or the California Institute for Regenerative Medicine. I'm going to talk about, in particular, the last one, age-related macular degeneration. And when you think about stem cells, I noticed just recently we saw for the first time pictures of Pluto, and the space program is amazing. It's a complicated program that involves interdisciplinary science and actually people in clean rooms building machines. Well, stem cell therapy involves also people in clean rooms, but instead of making the Mars rover, they're making cells. And they have to do it such that no bacteria or viruses contaminate the cultures. And in some ways, there are similarities between stem cells and space. They're both high-risk, sort of high-gain activities that cost a lot of money, but have great potential for letting us learn about different things. And I like to always start, when I talk about stem cell transplants, to talk about the challenges in stem cell transplantation. There's a lot of hype in the field and a lot of excitement, but we have to remind ourselves of the difficulties that we might encounter in doing this work. First of all, it may be difficult to obtain a suitable population of cells for use in therapy. Let's say you want to make a particular complicated type of cell. Well, it may be hard to get a pure culture of those cells. You have to somehow convert your undifferentiated stem cell into the cell of interest. Second, the cells may not survive, integrate, and function, especially in cases where the pathology is advanced. We may be asking a little too much of a cell to just squirt it in there and fix everything, especially if, for example, there's a lot of scarring and damage that's already occurred. So that's a consideration. And then third, the cells might be rejected by the immune system. And I mentioned iPS cells might be a way around this, but still a concern, and especially if they go to the wrong place. And then finally, this is one the FDA is perhaps most concerned about. The cells could grow out of control. If you take an undifferentiated cell, remember, one characteristic is it likes to grow, replicate, make more copies of itself. Well, we don't want to put a cell into a patient and have it grow and form a tumor. So we have to be careful of all of these points. And also, you have to be aware of snake oil. And if you look on the web, you can find all kinds of stuff. Unproven, unregulated stem cell therapies that are preying, companies that are preying on desperate patients. I have up here foreign stem cell companies, but even in the United States, you can find pretty shady activities going on. So you have to be really careful. And actually, there are links on the CIRM website that can allow you to check out a company if you're interested in knowing if it's legitimate or not. Now, that all being said, the eye actually is a good place to start in developing stem cell therapies. There are advanced surgical methods to get into the eye, into the back of the eye, and the retina, which I'm going to talk about in a minute. They're non-invasive imaging methods where you can see the different layers in the eye without cutting into the eye, just by looking from on top. And they're good endpoint parameters to tell if therapy is working. If you think about treating spinal cord injury, they inject billions of cells into the spinal cord. They can't follow them. They don't know where they go. And it's very complicated to tell if it's working. Unless the patient gets up and starts dancing, which probably won't happen, there are very subtle changes that might occur. Well, in the eye, not only do we have eye charts that we can have the patients look at, but we have very advanced methods to tell and map out the visual field and tell where the photoreceptors are working. And then finally, we don't think we're going to need billions of cells in the eye. We think we can put in a smaller number of cells, maybe on the order of 100,000, and really see a difference. So it makes the eye an excellent candidate for developing cellular therapies. And we lack treatments for ocular disease. The eye is a beautiful thing, but there are diseases like retinitis pigmentosa, Usher's syndrome, glaucoma, and macular degeneration. And you may know people that have these diseases. For example, Judi Dench has macular degeneration, and Lily Wash and Megan Downing have different kinds of diseases where they're losing their sight at a very young age, retinitis pigmentosa and Stargardt's disease. I was just at a conference in New Orleans for families of patients with Usher's syndrome. That's a genetic disease, and the patients are born deaf, and then they get retinitis pigmentosa and go blind between the ages of 16 and 30. So a real awful, horrible thing that maybe we could treat using stem cells. And that's the whole idea of regenerative medicine. Can we treat disease by replacing damaged or dying cells using cell therapy? So what cells do we need? Well, let's delve into the eye a little deeper and look at the tissues that we would need for these different diseases. And if you look in the back of the eye and cut a section through the eye, it's kind of like cutting a section through a cake, and there are different layers. And the retina is the material in the back of the eye about as thick as a wet piece of tissue paper that folds around the eye, and it has these different layers shown here in this cartoon. If we're talking about retinitis pigmentosa, in that disease, the rods die. And you start by having night blindness and tunnel vision, and then eventually the cones in the middle of the retina die. So rods and cones are disappearing. So you would need to make those cells to treat that disease. In Usher's syndrome, same thing. It's basically a form of retinitis pigmentosa. In macular degeneration, it's a little different. The cells in the back of the eye, the RPE, retinal pigmented epithelium, dies first. And the RPE is a crucial support cell for the rods and cones. So when RPE die, pretty soon the rods and cones die. And it occurs in the macula, which is the central part of the retina, important for high acuity vision. You can actually see the RPE if you look at somebody and you look at the eye, the pupil is black. It's not because it's dark in the eyeball. It's because there's a pigmented monolayer behind the retina that's darkly pigmented, and that's what the RPE is. And then finally in glaucoma, the retinal ganglion cells down at the bottom of the picture die. And the retinal ganglion cells are the important cell that sends the information to the brain via the optic nerve. They send these long processes called axons down the optic nerve to the brain. And often associated with high pressure in the eye, those RGCs die. So tall bill to fill in different cells needed for different diseases. And then if you think about late stages of macular degeneration or Stargardt's disease, you would need a combination of cells. You need RPE plus photoreceptors, sort of a bilayered sandwich to replace that part at the top of the slide. Another approach that people are trying is one called neuroprotection. And the idea here is, let's just put in a cell that secretes a lot of growth factors and maybe keep the photoreceptors alive and prevent them from dying. And perhaps a support cell that could make growth factors would work to keep RPE alive, or photoreceptors alive, or retinal ganglion cells alive. And that's being tried as well. Okay, so how can we make these cells? This is a complicated structure, and they're all very different. They express different gene products. How do we make these cells? Well, pluripotent stem cells, both ES, embryonic, and IPS, can be converted to ocular cells, and that has people very excited in the field. Maybe we can treat these diseases. Here's just an example. This is the cover of Nature showing in green a structure that was seen in a dish in mouse embryonic stem cells. And it looks a lot like a developing eye in an embryo. And Yoshiki Sasai went on to show that indeed, you make what are called optic vesicles, little budding eyeballs staring up at you in the Petri dish under the right conditions. And David Gamm at the University of Wisconsin has shown that you can convert these to make structures that have all of the layers in the retina, including the photoreceptors. So there's a lot of excitement about making retinal cells from stem cells. Our lab is focused on the RPE, and the work of Brittany Pennington and Roxanne Crows and others in the lab has shown that we can take embryonic stem cells or IPS cells, and under the right conditions, we see pigmented cells popping up, and we can purify these and get cultures that are 99% positive for RPE markers and very homogeneous. So we've teamed up with David Gamm to combine RPE with photoreceptors, and we're funded by the Foundation Fighting Blindness in what's called a WIN-GUN Translational Research Acceleration Program grant. And the idea is make a monolayer of RPE, add photoreceptors on top, and use that combination to treat what I mentioned before, late Stargardt's disease or atrophic macular degeneration. So combination of cells. And we're just getting started on that research. I want to end, though, by talking about a project that's a little further along, and that's developing a cellular therapy for the dry form of age-related macular degeneration, or AMD. Now, there are two forms in this disease, the wet form and the dry form. The wet form, it's only about 10% of the cases. You can actually get a shot in the eye to block blood vessel growth, and that works pretty well for most patients. The dry form, unfortunately, there's no good treatment. And in that disease, we know the RPE cells degenerate, which leads eventually to the death of the photoreceptors. So the idea is, let's go in and replace the RPE before the photoreceptors die and preserve the photoreceptors that you have. And in fact, studies in rodent models and in some patients in experimental surgery show that if you can put in fresh RPE into the macular area, you can preserve photoreceptors. And we know, as I said earlier, we can make RPE from pluripotent stem cells. So we've teamed up with an all-star cast of characters from the state of California and beyond to form what we call the California Project Cure Blindness. It's funded by our CIRM, California Institute for Regenerative Medicine. And the principal investigator is Mark Humayan, who's a retinal surgeon at the USC Eye Institute. And here at Santa Barbara, our job was to figure out how to make the RPE cells and develop that into a therapy. We've teamed up with Caltech, who developed a scaffold to grow the cells, and City of Hope, where they have a GMP facility, good manufacturing process facility, to make cells for clinical use. We've also partnered with University College London, where they have the London Project to Cure Blindness, and a startup company called Regenerative Patch Technologies. We got a grant in 2010 to develop a monolayer of RPE on a scaffold, and that's our approach. Imagine a little 3x5 millimeter piece of plastic, sort of like contact lens material, that's coated with a layer of cells. The idea is the surgeons would then go in and implant that in the back of the eye to replace RPE cells that are dying. The scaffold designed at Caltech is made of a substance called Parylene, which is already approved for use in the eye and is very biocompatible. And we've put this into rats, and you can see in the picture on the right, a blow-up, the parylene appears as a white layer with the human cells on top. And we've worked out a surgical procedure that we're now going forward with, and it would work like this. A slit is made in the side of the eye, the vitreous gel in the middle of the eye is removed, and then a little fluid is injected to separate the retina from the RPE. And a little blister forms. Then another cut is made, and the patch with the cells on top is inserted with about 100,000 cells on this 3 by 5 millimeter patch into that blister. Let's see if I can get that to go. Okay, there it is. And we've worked out the surgical procedure in the Yucatan mini pig, which has an eyeball about the same size as a human, and surgeons practice on that. And what you're seeing is an actual movie of the surgery taken by Rodrigo Brandt and Mark Humayan at USC. Here they're removing the vitreous gel, which is kind of a stringy material inside the middle of the eye. There they're creating that blister by injecting some fluid. And instead of a large slit, we've actually worked it down to a small hole that is cut in the retina. And here's our scaffold with the cells. It looks kind of like a wine bottle. And we developed this tool where we can pull that inside a cannula using a forceps, and it folds up kind of like a taco shell to protect the cells. And then that's inserted through the side of the eye into that hole and delivered into the subretinal space in between the RPE and the photoreceptors. Now, I don't know about you, I get a little nervous when I see sharp objects near my eyeball, but surgeons are pretty comfortable with this procedure, and we think we can train retinal surgeons to do this such that it can be completed in an outpatient procedure in about an hour. Okay, so let's see if we can go forward. So looking forward, and let me wrap up here, looking forward, like this guy is looking forward. Actually, you young people that are going to become ophthalmologists, this is really a bad idea. You should not have a sharp object in the eyeball of a child and be looking forward at us. Okay, don't do that. But looking forward, a lot of people are going forward with this kind of therapy. There are groups in Massachusetts, Israel, the Bay Area, Japan, New York, Washington, and London that are all developing therapies to treat the dry form of macular degeneration in various stages of clinical application. The group in Japan has already used iPS cells for the first time to make iPS RPE. And at the bottom, our California project, Cure Blindness, has just received permission from the FDA to go forward in a phase 1 clinical trial. So if you think about that analogy of the space program and stem cells, here's a picture of the improvements in the Saturn V rocket from 1967 and 1973. I think we're at a very early stage in stem cell research. Some things are going to work well, other things may not work, there'll be setbacks, and we need to keep developing therapies and learning from these advances. So I'd like to stop by thanking the California Project to Cure Blindness and the collaborators that are involved there, Mark Humayan, David Hinton, Larry Couture, Jamie Thompson, David Gamm I mentioned, and especially people at UC Santa Barbara that have contributed to the research listed here, especially Sherry Hikita, who led the project early on, and my collaborator, Link Johnson. And finally, thank my funding sources, the Foundation Fighting Blindness, the California Institute for Regenerative Medicine, and the Bose Foundation. Thank you very much for your attention.
Hello.
Hi.
Okay. Hello. Thank you. Since this study is still under development and it's very young, what would be the cost of a cell transplant like the one you showed us in your presentation?
What would be the cost? That's a very important issue, and one of the things that we've talked about at the California Project, we would like to keep the costs down and make it affordable to patients. We're hopeful that it would just be a one-time treatment, an implant that would last the rest of your life. Unlike the wet form where you continually get injections every month and it's very expensive, we haven't worked out the exact cost, but We know that we can make these cells and we can make this scaffold relatively cheaply, so we're hopeful that we can keep costs at an affordable rate that insurance will cover.
Okay, thank you.
Sure.
Earlier when you were talking about turning like skin cells and then turning them into iPS cells, does it affect like the rate or like the rejection? Like, is there a lower level of rejection when you do that method as opposed to like another one?
Yeah, so that's the idea, is that you could use a patient's own skin cell. Let's say I get Alzheimer's disease. I could take my skin, convert it to an iPS cell, then convert that to a neuron, and then maybe treat myself. And theoretically, at least, it would be matched to my immune system, would not be rejected. And they've just reported a couple of different studies in mice suggesting that that may be the case. And there's some ongoing work in monkeys now to test it in an animal model that's closer to humans. But here's one problem with the iPS approach. Let's say you have retinitis pigmentosa, and it's due to a mutation in the gene encoding rhodopsin. Well, now the skin is going to have that same DNA. It's going to have that same mutation. So if I make photoreceptors, it's going to be a problem. So in that case, you have to have an additional step in there to correct the gene mutation in those cells, or use a bank of cells that are matched to all the different people in the HLA locus. And the idea is you could then, sort of like blood typing, go and find a cell in your bank that matches the patient, doesn't have any mutations, make the cells from that and put those in.
Thank you. Hi. So
Hi.
kind of similar to the last question, I was just wondering, you mentioned that like the first stem cell therapy was bone marrow transplants, and I know that with those, it's sometimes hard to find a person that can match, or I don't know, to find a person that can match you, and so your immune system doesn't attack the cells. Is that true of ocular cells as well?
Well, yeah, good question, and of course we have a diverse population here in the United States, but several groups are making banks that they hope will match in at least several of the HLA loci that determine rejection. And for example, the group in Wisconsin I'm working with is making a bank of, they think about 200 cells that will be matched in 2 of the major histocompatibility loci, which will be a pretty good match and they think would be useful in transplants. So the advantage of having that bank is that you walk in and the cells are all there ready to go. You don't have to wait and make iPS cells. In the case in Japan where they made RPE cells for macular degeneration, it took about a year. They had to take the skin. They made 26 lines or so of iPS cells. They screened those for making RPE. They did a number of tests, and it was a year until they were ready to put the transplant in. So, and it was very expensive. And that's the problem with autologous as well. It's very expensive endeavor. The Wall Street Journal estimated that it would cost about $512,000 per patient to make a particular cell type from iPS from the patient. So a lot of people think that it's better to have a so-called matched bank that could be used off the shelf.
Thank you.
Sure.
Okay. When you guide the stem cells to differentiate into different cells, and you're doing it for like a full organ or like an entire eye, do you have to do it layer by layer for the different kinds of cells, or?
Good question. It varies from cell to cell, and it's sort of the holy grail in the field. You know, you have to figure out how to make the cell that you want. In the case of the RPE, we took a couple of years to figure out a protocol, a method in the lab where we could get 99% purity. And in the case of the photoreceptors, we're a little further behind. We can't get there yet. And they developed these structures that have the layers, and it just happens. It's like the cells are trying to make an embryo in the dish. And they're not mature, though. They're not a complete eyeball. It's not like you don't actually see an eyeball looking up at you in the dish. And so we're still developing methods, and it's a challenge. You try to add the media in the dish that encourages the cell to turn into what you want. And it's based on studies of model organisms like mice and frogs and worms and flies, where they've discovered growth factors, for example, that tell an early anterior neural plate cell to become early eye field, which then becomes eye, and then one path goes to RP, another path goes to retina. And you try those in the dish and do studies to tell how pure they are. And it's a long process, long, expensive process. RPE, I think we're there. Photoreceptors, not quite yet.
Okay, thank you.
Sure.
Thank you. I think my question's relatively simple. I just
Okay.
was wondering, why are we going with skin cells? It seems like, as you implied, they would be the ones with the highest rate of mutation. Is it just like a matter of accessibility?
Good question. Matter of accessibility and protocols were developed there first. I guess the reason is Yamanaka started with the skin of a mouse, and so we started with skin of humans. They can now do this from blood, which is a little easier for the patients. It's a little more difficult. It's not as efficient from blood, but methods are getting better and better. And yeah, I think it depends how many cells you want, and maybe the methods, I think, are going to improve and make that an easier process.
Thank you very much.
Sure.
Earlier you were talking about animals, that you were testing on animals, but wouldn't it have different reactions on the human body?
Ah, yes. Well, that's a real problem in developing drugs of all kinds. You know, you hope that the animal model is going to reflect what's going to happen in patients, but a lot of times it does not. And the reason is that a mouse is a mouse and a human is a human, and there are differences. What you have to be able to show, first and foremost, to get into a clinical trial to test on humans is safety. And the mantra is do no harm. And so the FDA really wanted to look at our data to make sure that it wasn't toxic to the rats that we were putting in or the pigs that we tested on. And in fact, in the case of macular degeneration, there really isn't a good animal model that reflects all aspects of the human disease. The macula is an area full of cones, for example, which is used in normal daylight vision. Rats are nocturnal. They actually don't have a cone-rich area in the middle of their retina. But the model we used has a mutation in the rat that makes the RPE defective. And so we could at least ask, can we replace the RPE and get RPE function in the rat? But you bring up a very good point, and a lot of drugs fail for that reason. They work in animal models. You get to the human patients, you try it, you spend lots and lots of money, and then it doesn't work. But you learn something. Going into humans, even if the drug fails, you learn something. So it's a difficult process, but we have to keep trying.
Hello, as a follow-up to the last question, I was wondering, have any potential treatments failed on animals and therefore haven't been implemented on humans that could actually be just specific to that animal? Like,
Good question. Have studies failed in animals and for that reason never made it to humans? You know, I guess you can't answer that because they never get approved to be used in humans. And it could very well be that that might be the case, you know. Maybe something doesn't work because the animal's different, and the problem is you then can't get to the human to try it. So, good question, though.
thank you.
Okay, let's thank Dr. Clegg with a warm round of applause. Thank you.
Thank you. Thank you. Thank you.